Hi everyone, thank you for joining today's Coffee Science Seminar. It's a pleasure to facilitate this presentation of Shanice Van Heften. I'll just introduce myself. I'm Dr. Millicent Smith. I'm a lecturer in crop physiology here at the University of Queensland. I'm jointly appointed between the School of Agriculture and Food Sciences and the Centre for Crop Science in Coffee. And it's one of my greatest joys to be a supervisor for Shanice, and it's a pleasure to facilitate this seminar today. Before we get started, I'd like to acknowledge country and really acknowledge the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country and recognise their valuable contributions to Australian and global society. Just some housekeeping things for today. So the seminar is scheduled to start now and we'll finish by 1 o'clock. Shanice will talk for about 45 minutes and we'll have some time for questions at the end. Please use the question and answer box, not the chat function. We'll go to that at the end for any questions that you might have to answer. So we can get to those at the end. So Shanice Van Heften is a PhD student at Coffee. Her PhD research focuses on using new innovative tools to increase the productivity and reliability of mung bean production for Australian growers. Her research is supported by the GRDC, the Grains Research and Development Corporation, through a research scholarship, and the Australian Centre for International Agricultural Research through the International Mung Bean Improvement Network. Prior to commencing her PhD, Shanice completed a Bachelor of Science and Bachelor of Business with honours at QUT. We try not to hold that against her too much. And she's passionate about improving global food security and using her diverse backgrounds and skills to be part of developing solutions that will sustainably feed communities into the future. And I'm sure that if you're hearing from her for the first time today, it won't be the last. She's going good places in the future. So I'll let Shanice take it away and come back to you at the end for any questions you might have. Thanks, Shan.
Thanks so much, Millie. So as Millie mentioned, I'm Shanice Van Heften and I'm a PhD student here at Coffee in the Centre of Crop Science. And today I have been given the really fortunate opportunity to be able to present some of my research to you guys. And this research is titled Understanding the Genetics of Mung Bean Senescence Behaviour Using Longitudinal UAV Data. But before I start, I too acknowledge the traditional owners and their custodians of the lands on which we meet today and pay my respect to their ancestors and their descendants. So I thought I would start my presentation by providing some background into what is mung bean and why is it important. So for those who aren't familiar with the crop, it's a short duration tropical legume, and depending on the cultivar and environment, can reach maturity anywhere between 55 and 110 days after sowing. And it's because of this short duration that it's a really valuable choice for double and intercropping systems, as it's short enough that it can fit really nicely in between other major crops like wheat and sorghum. And it's also an important source of dietary protein and nutrients, in particular for countries where they're mainly cereal-based diets. Mung bean provides a really nice option to supplement these diets. And lastly, mung bean is predominantly grown across Asia and Africa, and so it's a really important source of income for a lot of these smallholder farmers. So as I mentioned, mung bean is mostly produced in Asia, with the top producers being India and Myanmar, but we are seeing that a lot of other countries are starting to produce mung bean in larger amounts, in particular Australia. And so mung bean is now cultivated on over 6 million hectares of land globally. So if we deep dive more into mung bean production in Australia, it's actually one of Australia's highest value grain crop. So mung bean is produced in northern New South Wales and southeast Queensland. And you can see from this figure here that in the last 30 years, there's been a steady increase in total mung bean production. But it's pretty evident that we do get quite variable yield production. But I guess the thing that is most consistent is its worth, with it being consistently worth over $1,000 per tonne. But there is a real potential for this production to increase as we start looking at Australia's plant-based protein market. So these values here are from a Deloitte study that is really highlighting the current market in Australia. So currently in Australia, we produce around $185 million of consumer expenditure on plant-based proteins and have around $2.7 million in export. But by doing some predictions, it was seen that these numbers explode by 2030, with expenditure increasing to 3 billion and exports increasing to 338 million. Another trend that we're seeing is that a lot of Australians are eating less meat, with 1 in 3 Australians consciously limiting their meat consumption and looking for alternative plant protein sources. So this means that there's a growing need for looking at alternative plant protein sources. And so there's an opportunity for Mungbean to really take a piece of that pie. However, a big thing with Mungbean is that its expansion is limited due to its low yield production and variability, which we really saw in that graph before when we looked at total production. And so when we look at research plots as well as in simulations in AppSim, we can see that Mungbean has a yield potential of over 2 tons per hectare. But we commonly see with growers, they can produce anything between 0.5 to 1.5 tons per hectare. So there's a really big gap between potential and actual yield. And the reason for this is due to a number of things, in particular, Mungbean being quite vulnerable to a range of abiotic and biotic stresses, as well as this crop displaying a range of unfavorable traits for large-scale intensive cropping systems. And so the trait in particular that I'm interested in is its asynchronous flowering behaviour. So essentially what this behaviour means is that the plant will produce flowers in multiple flushes, and this then leads to the plant having a staggered maturity, which can create a lot of challenges when harvesting in both hand harvesting and mechanical systems. And also it means that the plant will continue to produce biomass after flowering, which creates this competition between resources from the leaf biomass to its yield components. So you can see it's quite an extreme case here, but we do see this in the field. On a plant, we can have mature pods, flowers, and just developing pods at once. So this is a real issue when it comes to, as I mentioned, challenges when harvesting. So I guess I wanted to kind of frame that challenge in terms of what Australian growers deal with. So imagine you're a farmer and you have your Mungbean field, and you need to determine what is the best time to desiccate the crop for harvest. So with Mungbean, we need to apply a desiccant to essentially just kill off and dry off that biomass that's accumulated in order to harvest. So imagine you're the farmer and you walk into your field. You need to look in your field and determine whether it's the right time to spray. And as you can imagine, it can be quite difficult if you're seeing a lot of biomass as well as a lot of flowers and green pods and developing pods. But you make the decision. Obviously, it's not that easy, but once you make that decision, you then spray the field with the desiccant, and then all the biomass will senesce and you'll be ready to harvest. And this image here is how we harvest Mungbean. But as I mentioned, it's quite difficult to determine that right time. So imagine you have 2 scenarios and you desiccate. If you desiccate too early, it can result in some of these green pods that it's still developing, never being realized as grain yield. And there's also a risk of high chemical residue on seed. So we're seeing in Mungbean, a lot of research is coming out that if you spray too early, it can lead to high chemical residue on seeds. And with there being a growing restrictions on seed residue levels, that's a real issue that a lot of growers are going to have to deal with. Whereas if you spray too late, a lot of these really mature pods are at high risk of shattering. And you can also have risk of post-spray sprouting. So you pretty much desiccate the plant and then then it might rain and then the seeds will start to sprout in the pods. And that was a big issue that I had last season with all of that heavy rainfall. So I guess this raises the question, can we design the ideal Mungbean crop? And so thinking of this challenge, it could potentially have synchronous flowering behavior, which can then lead to synchronous maturity. And it can also have an earlier onset senescence, which means that the plant is able to effectively take those resources from the leaves and put it into yield components while reducing overall biomass, helping to regulate some of that desiccant use. However, another thing to consider with mungbean is that a lot of these current varieties have a really narrow genetic base. And so compared to a lot of other major legumes like soybean and chickpea, genetic improvement has been slow just because it's quite new, but there is 43,000 diverse mungbean accessions available globally. So that means there's a real potential to explore genetic variation in order to increase yield, productivity, and reliability. And although mungbean is the new kid on the block compared to the other legumes, there has been a really massive global effort for mungbean improvement. So not only in Australia, where we have a really great mungbean improvement program at DAF, but there's also been investment by ACR for an international mungbean improvement network. And so the aim of this network is to increase farmers' income, systems productivity, and food security by delivering high-yielding, disease and pest-resistant mungbean varieties to smallholder farmers. And so you can see in this image here that there's a lot of partners across Asia as well as Africa, and Australia is also a part of this network as well. So we're kind of in this really exciting time in agriculture where we have a lot of new technologies and methodologies that have been developed to help accelerate crop improvement. And these have really been applied to a lot of major crops. And there's an opportunity to therefore apply them to mungbean to really streamline this breeding process. So I've just listed some of these tools and methodologies in this process. And so for mungbean, we're really lucky that we have a lot of diverse germplasm available. And a lot of these collections have been genotyped through the network and by the mungbean improvement program, which is great. But it does mean we have a lot of genetic information, and we now need to kind of translate this information to physiological and characteristics that we see in the field that are of value for farmers. So connecting the genotypic information to the phenotypic information is a challenge because we really have a phenotypic bottleneck, which I'll describe in my later slides. So that's when high throughput phenotyping platforms, and in particular UAVs with sensors, could really offer an opportunity to kind of bridge that phenotyping gap. And currently there hasn't been any research published in using UAVs to phenotype technology, to phenotype mungbean. So once we can really optimize this beginning process of the pathway, we can then start looking at these other cool methodologies that can help really accelerate improvement to develop mungbean cultivars with higher productivity and reliability. So that brought me to my project question, which asked the question, can we use UAVs to phenotype mungbean canopy traits? So I'm really lucky for my project. I get to use a mungbean nested association mapping population, which was developed by QUT as well as by the mungbean program at DAF. And so the way this population was generated was selecting 30 diverse donor lines from a diverse collection, and we crossed them with an elite commercial cultivar crystal. And these diverse donor lines consisted of germplasm, other commercial lines, as well as some wild lines. And it generated a population of around 2,060 accessions. So you can see from this image here that we not only see quite a lot of diversity and variation in seed color and size, but when you look at the canopy, there is a lot of variation there. So it raises the question of whether we can exploit some of this variability to improve productivity. And so to answer this main question, we then broke down this question into 3 objectives. And so the first is understanding what physiological traits have a relationship with UAV traits. So it's all good that we're able to get this information from the UAV, but what does it actually mean in regards to what we're seeing on the ground? So I looked at data from 2 field trials, one in Warwick 2021 and one in Allarat 2022. And these trials had a subset of the NAM population sown, so 6 pedigrees, and as well as the NAM parents. And so the way we acquired the UAV data is we flew our drone, which had a sensor on board it across the field. regularly throughout the season. And so how this works is that the drone will fly slowly across the field and take individual images as it's flying across. And so with these individual images, we then stitch them together to create an orthomosaic, which is essentially just a large image of the field. We then create a shapefile, which segments out each individual plot in the field. And from that, we can extract that UAV data from each individual plot at each time point. So these data points are usually vegetative indices, which are created based on reflective bands in the image. And so some of these indices that we look at are OSAVI, NDVI, and NDRE. And so these have been linked in other crops to be associated with biomass, vigour, plant health, and chlorophyll content. We also have canopy coverage, which is just a proportion of canopy coverage within the plot, and thermal, which is linked to canopy temperature. So alongside these UAV data points, we then collected a range of ground-based measurements. So some of these included Fusarium wilt scores, which although was definitely not intentional in my trial, but it's a really nice way to showcase how UAVs can be used for disease screening. We also looked at early vigour, total above biomass at multiple time points, as well as a more leaf level trait, which is stomatal conductance. So in terms of Fusarium wilt, so as I mentioned, it wasn't an intended infection in my trial, but it did happen. And Fusarium wilt is becoming a really growing concern for growers and can lead to devastating yield losses, so up to 100% in yield losses. And so the way we captured this was through a visual score, with 1 being that there was no symptoms in the plot, and 9 being that all plants were impacted and pretty much dead. And so to capture this measurement can take between 3 to 4 hours, depending on your experience. But obviously being a visual score, it means that the scores are quite subjective and they are less quantitative because it's just 1 till 9. And so when looking at the wilt score compared to the UAV traits, we did find a significant relationship with the vegetative indices, but not with the thermal variable. And so essentially, if there's a high vegetative indice, it was linked to the plants that had no symptoms, so able to accumulate high biomass and greenness. And so I guess why this is really cool is because to fly the drone across the field was only 20 minutes compared to the 3 to 4 hours. And you kind of lose that subjectivity because you're getting data from a machine. And it also means we can look at a lot more variation within the plot instead of just a 1 till 9. And often at times with a lot of disease screening, they capture a measurement at 1 or 2 time points. But because you can screen a large area really quickly, it means that researchers and breeders can start screening across the entire season. And for a disease like Fusarium wilt, it might look like a plant is fine at 29 days after sowing, but at 50 days, it might be completely dead and impacted. So it's really important to understand how progressive the disease is. The next we have early vigour. So early vigour is linked to establishment, rapid canopy development and yield. So similar to the disease scores, we did a 1 to 9 system, with 1 being the most vigorous and 9 being the least vigorous. And it has the same limitations that I mentioned with Fusarium. So it takes between 3 to 4 hours. They are subjectives and they're less quantitative. So we did that at both Warwick and Allarat, and we saw the same trends and relationships across 2 seasons, which means that all of these indices are quite good and stable across multiple environments. And I do have a trial this year as well that I'll be analysing, but from preliminary results, it looks like we have the same trends as well. So it just highlights that it's really easy to use this technology across multiple locations to look at the same trait. And so from these relationships, we can see that if the plot has a high vegetative index, it means that the plants are most vigorous. And with the canopy temperature, if there's a high canopy temperature, it means that the plot is least vigorous. So the next trait is stomatal conductance. So stomatal conductance is linked with crop productivity as it's associated with water use. And so we actually use this piece of equipment here, which is a handheld device, and it's actually considered a high throughput piece of equipment compared to its predecessor. And so even though it's good because you can quickly take it in the field and quickly capture measurements, it can be quite variable and time sensitive. So if you take this measurement on one part of the leaf, it could be different if you took it on another part of the leaf. And also because stomatoconductance is driven by environmental conditions, if you took a measurement at 9 a.m. and then you came back and took it at 12 or had to walk across the field to another plot and it takes a few hours, it means that you are impacting your results. And so for our study, it took us 2 hours to do 33 genotypes for 4 reps. So that was around 120 plots, which isn't too bad, but we are limited by the number of genotypes that we can explore. And so when we did this in Warwick, we didn't see much of a relationship between some of these indices, but what was really nice is that we did see a relationship between the thermal and GSW. So that means that when we have a high stomatoconductance, it means we have a lower temperature, and with the lower stomatoconductance, we have a higher canopy temperature, which is what we would expect, because as the stomata closes, it increases internal temperature. But it's really neat that we can see this relationship with the UAV, particularly with just a multi-spec sensor, because it's considered not as high resolution as some of the other sensors you can get. And so it would be a really valuable tool in breeding. So what does this mean? It means we can potentially start exploring the water use efficiency of genotypes, and we can also start looking at how sensitive the stomata is to changing environmental conditions, which is particularly important if you want to look at things such as heat stress as well as drought stress. So it really highlights the potential of using this technology for some of these more detailed leaf level traits. And the last ground-based measurement is total above ground biomass. So biomass data, as we know, is important to understand light interception as well as assimilate partitioning to yield. So a major limitation of capturing this trait on the ground is that it's destructive. So we have to take out that part of the plot in order to do the hand dissectioning. So in our case, we did around 66 plots, and it took us over 9 days to harvest, dissect, dry, and weigh samples. And therefore, it means you can only really look at a small number of genotypes, unless you are a large breeding company with a lot of people who can help, but realistically, that's not the case. So yeah, you are quite limited in the number of genotypes you can examine. And so in Warwick, we see that there is a significant positive relationship with all the vegetative index, which means that high vegetative index means higher biomass. And there's also that same relationship with canopy temperature. So high canopy temperature, lower biomass, which could be linked to stomatoconductance or could be linked to a cooler canopy because the canopy is closed up in the plot. But it's an interesting way to start exploring some of these relationships. And so just for simplicity, I just showed OSAVI, which is the key vegetative index that I will be looking at in the next section. But in ALARA, we do see a positive relationship at 50% and 90% flowering, but the relationship for maturity, so 90% black pod, isn't as strong. And I'll explain why later on, but essentially it kind of highlights the fact that we may need to look at a more complex approach when exploring some of these indices at different time points. So now we understand if we take UAV data, what is that linked to on the ground? The next question is, can we then use UAV to explore canopy growth over and senescence over time? Because a lot of these ground-based measures that we looked at before was at a single time point. But as I mentioned with the UAV, we can start capturing data once a week, twice a week, as much as we want, so we can get this really nice look at how the plant is behaving over time. So for this research objective, we will look at the ALARA trial. So we captured a drone flight every 2 weeks throughout the season. So there's around 9 flights in total. And alongside that, we just took detailed phenological measurements. So we have UAV data for each time point for the entire field. So the first thing we did was undertake a two-stage approach. So the first stage involves fitting a spatial model to the phenotypic data separately for each measurement time. So this allows us for each time point to reduce some of that variation that may be due to experimental design. And that gives us an adjusted value for each time point for each genotype. And then with these adjusted values, we can then fit a P-spline across each time point. And so that gives us a really nice growth curve for each genotype that we can start dissecting further. So for each of these curves, we can then pull out some of the growth curve parameters based on their predictive values, as well as their first derivatives. So if we look at early canopy development, we can start looking at the rate of canopy accumulation and at what stage it reaches its maximum canopy growth speed. We can also look at a trait called area under the curve, in particular proportion of area under curve before peak. And for those who might not be familiar with area under the curve, it's essentially a metric used to look at the area in accumulation under this part of the curve. And then we have our senescence traits. So we have our peak vegetative index, and when it reaches that peak, we have the proportion of area under the curve after the peak, as well as how much reduction in the vegetative index from this peak to 90% black pod. So there's a range of components that we can start analyze and dissecting in order to see whether there are particular traits of interest that we might want to consider when trying to improve Mungbean productivity. So looking at the growth curves with just the parental lines, we can see that there is not as much variation at the start, but as we start furthering in development, we're getting a lot more variation across the genotypes. And what's pretty cool is we see at these two time points, there's really a bit of a slowdown in growth. And so we found that in Alara, so in Queensland, we had a lot of wet weather events, and in Alara, we had two major weather events of rainfall at these two time points. And so we're seeing that although there is accumulation in heat units, we're not seeing growth, which may be due to a range of things, the waterlogging, cloudy sky, as well as temperature. And so it really slowed down growth. And then we can see that there's an increase back in growth. So it's really nice that the drone is able to capture some of that canopy dynamics. And when we look at the families for the NAM population that we explored as well, we see two interesting things. The first is that some of these families that are crossed with crystal and another commercial line, they have very little variation in their canopy dynamics and their growth curves, especially when you compare to these families, which are crossed with the commercial line crystal and a germplasm line, where there is a lot more variation. So this highlights that there's really is variation in that we can explore in this population, and we can start understanding whether some of this variation could be useful in improving productivity. Another thing that was interesting was that this peak that we're seeing for all of the genotypes, they all are reaching around mid-pod fill. And so this supports a finding from Gitika, who was a PhD student also at QAFI. And in glasshouse experiments, she was able to identify that mid-pod fill is the key transition from vegetative to reproductive components. And this supports it, but at a larger scale. So I thought that was quite interesting. And we're also seeing some of these slowdown components as well. And so when we pull out some of those traits that I mentioned before from the growth curves, I just wanted to display here that there is some variation for a lot of them. And so this red line here is where crystal sits. So there's a lot of diversity that we can really tap into for these traits. So I guess that part of the research question really highlights that we can use UAV to start looking at different traits. And that's great, we can find variation, but that doesn't really matter for breeders unless we can ensure that there's a genetic basis underpinning them. So for the final objective, I wanted to really explore if we can dissect the genetics of some of these UAV traits And so I just wanted to look at some of these senescence traits as a bit of a case study. So the peak OSAVI, the reduction in OSAVI from the peak, and the proportion of area under the curve after the peak. So from doing a GWAS, undertaking a farm CPU approach, we looked at the 3 traits, and we can identify that there are significant QTL associated with them. But I guess the key thing that we want to know is, are any of these QTL linked also to phenology? Because obviously we want to find QTLs that are independent, because we don't want phenology to be able to influence these traits. And so when doing a GWAS on some of these key phenology traits, we can see that there are some that are shared among them, but there are a number of other QTL that are associated with these traits as well. So that means this really highlights the potential of using UAVs for breeding purposes. So we can able to rapidly phenotype large populations, and we can extract out traits that we could identify the genetics underpinning them and feed them into breeding programs. And so once we do that, we can start doing AI-guided breeding approaches, speed breeding, gene editing to further explore the functionality and utility of these genes for crop improvement. So I just wanted to finish my talk by highlighting some key messages. So the first is that UAV traits can be used to efficiently capture imported canopy and leaf level traits for Mungbean. So a lot of these traits like disease, screening, early vigour, biomass, these are really important traits that breeders do capture. And so using this technology allows them to capture this data in more detail. And another point is that this methodology can be applied to pre-breeding populations to dissect the genetics of the traits. So particularly for canopy development, it's a really hard trait to look at overall. So if we can really streamline this process and get some really valuable information, it can be then fed into pre-breeding programs. And finally, this methodology can be used to accelerate and support large-scale phenotyping in breeding programs. So just reiterating the utility of this technology to really help accelerate breeding efforts. So thanks everyone for listening. I'd like to acknowledge everyone who has helped support this project, in particular DAF, as well as the Unmanned network, as well as GRDC and ACR. Thank you so much.
Thank you, Shanice. It's a really nice talk, as always. We do have a question already in the chat, and I'll just remind everyone to use the Q&A function when they're asking their questions, and I can then read those and answer them. The first one I'd like you to answer, Shanice, is from Daniel Odwani. Did you consider genotypic differences in seed size in your early vigour measurements?
I have thought about it, but I haven't explored it yet. So that will be definitely something I will look at, potentially in understanding how the difference in these traits by clustering them by seed size and whether that impacts anything. But I guess I just wanted to see overall vigour.
And we do know there is interesting variation in seed size in this population. So it's a good question. Yeah, thank you. While we wait for any other questions, I was going to ask you, how feasible do you think it is for particularly our partners in the International Mungbean Improvement Network to use these sorts of approaches in their own programs?
Well, we're kind of finding that a lot of these technologies really are becoming cheaper as years progress. We recently got access to a new drone that is tiny and half the price of the drone that we're using, and it has the same functionality. So I guess it would kind of be like with everything, providing training and support to help facilitate the use of this technology. But then also, if certain partners use it and can feed it back into the program, because sometimes using these technologies aren't really the priority for some of these partners.
Yeah, it makes sense. So Bradley Campbell has asked, are the diseases you can phenotype limited to those with severe leaf symptoms?
Are diseases limited with severe limited seasons? Yeah. So I guess you would have to have some type of physiological growth impact from the disease. So if you've got a disease where it's just, and they would all impact the biomass components. So I guess it depends if there's a disease you think that doesn't impact the folia. Does it have folia symptoms?
Yeah, I guess you just need to be able to see the disease above the canopy, in the canopy, to be able to detect it. So it would be ideal for those sorts of diseases.
Yeah, and I find that the UAV is pretty sensitive to changes in biomass, whether that be from senescence, because you've got indices that work on the yellowness. There's also other components you can extract from the UAV, like height, as well as like a volumetric, so how big the plant is, so it's not an indice, so that can help further identify some of those differences that you might not see using just traditional vegetative indices.
So someone anonymous has asked, have you further looked at the closely associated genes with the significant markers that you identified from your GWAS?
No, I haven't. The only ones I looked at was the phenology, QTLs associated with key phenology stages, but it would be good to see how they are linked to what other people have found, hopefully other canopy development traits.
Yeah, good question. So RCN has said, interesting talk. Do you find any relationships between canopy structure and yield or yield-related traits?
So I guess if we're doing stuff on the ground, we do see relationships between just traditional ground-based canopy measurements and yield. For the UAV data, unfortunately, in that season, we weren't able to capture yield just due to some of those issues that I spoke about, the post-sprouting. But this year, we've actually collected a really nice data set with UAV data and yield. So it'll be really interesting to explore some of those growth curve parameters and whether they are linked to some of the yield traits that we were able to capture this year. It's been a tough year to do a PhD on Mungbean and get yield.
Yeah, so Joanne DiHaveri asked a similar question, which is thinking about relating those vegetative indices to final yield. And yeah, unfortunately, Shanice has had a bit of a rough time with her trials over the past few years. With it being so wet, we lost a couple of trials, and yeah, we lost the yield essentially of those trials, which was really challenging. But I think what you show today is that you've been able to get a lot of really interesting data from the trials anyway, which is really good. And we got yield from this season, so that's a good thing too.
And we can really link at UQ, there's been really great work on people looking at growth curves and growth rates in a glasshouse level. So if you consider the findings from that and explore the data we got, we can kind of start getting an idea of how potentially these components could link to yield at a smaller scale.
Yeah, hopefully you'll be able to do that with your data from this season.
Hopefully.
Okay, another really nice question. So from Cole Douglas, the canopy growth curves that you plot for each NAM population are really interesting. How much scope will you have to look into the specific traits, or are there plans for continuing the dissection of Mungbean physiology and yield beyond your PhD?
Yeah, so the goal now, since I have 3 seasons that I'll be able to do this longitudinal analysis, the goal over the next year is to be able to dissect that in more detail. We also get to use a really cool machine over in Taiwan, which does really detailed dissection of canopy that we can't do in Australia. And so that's looking at like 3-dimensional growth and pod development rates. So we can start exploring a little bit more detail that we can't capture using UAVs and see how they are associated with some of these UAV traits.
So another question around UAVs, do you think that they'll eventually be like a giant NIR and each curve of the spectrum will have a particular phenotypic meaning?
I feel as though, well, heaps of people now are doing hyperspectral stuff, and so I think they'll probably start developing even more specific vegetative indices that are able to be really linked closely to physiology. But I don't know, things keep growing all the time, things keep evolving, so potentially
Yeah, and another sort of more detailed question around what packages you're using and what are the things that you take to consider when you're formatting your genotype information.
Yeah, so in the slides that I presented, that is just based on the UAV data that I extracted, and something that I do want to do is actually incorporate the genetic information of the population into the analysis, so we can have a bit of a more detailed look into how the genetics cluster and impact some of those growth curves. For the genetic stuff, we just use GAPIT, so the farm CPU analysis in GAPIT. And yeah, I guess that's the main one for the genotypic information.
Hopefully that answers your question, Anonymous. If not, I'm sure you can send Shanice an email. She can give you more detail. Okay, so we still have a bit of time. If anyone has any further questions, we're happy to answer them.
I guess if anyone's interested in any of the future results that come out, because we've got some really cool things happening, including looking at multi-environment analyses, feel free to follow me on Twitter and kind of follow the journey of some of these results. Hopefully we can get them published soon for everyone to also get their hands on.
Yeah, no, that would be great. So maybe I can explain what's happening next week. So next week we have another seminar on Tuesday at 12 o'clock from Jessica Cartwright, who's a PhD candidate in the Centre for Nutrition and Food Sciences. And it's about empowering Indigenous communities to improve their health through nutrition and introduction of a low sugar carbonated beverage. Sounds really interesting. So come along to that. And just encourage people, if you haven't already signed up, to go to the Coffee Science Seminar webpage and make sure you're signed up so you can get alerts about each of the science seminars that happen every week. And you can also access video recordings of previous seminars there as well. So thank you everyone for joining today for Shanice's seminar. As we mentioned, if you have any information, please email either Shanice or myself, and you can definitely follow us on Twitter and Coffee as well to get updates about our research going forward. So thanks again for joining. Have a great day.